Our next presenter will be Lixte Biotechnology, Bas van der Baan.
Thank you very much. Thank you very much for the invite. It's great to be in New York. They say it's going to be a little better in the coming days weather-wise, but thanks, Matt, for highlighting this and getting the invite here. It's a pleasure to be here and talk about Lixte, a company that is developing a protein phosphatase 2A inhibitor for oncology. Lixte is also NASDAQ listed, and therefore we'll have our forward-looking statements here, and we just filed our 10K, so yeah, you can read the latest there. So my name is Bas van der Baan. I'm the CEO of Lixte. I took over from the founder in September last year. I've founded 2 molecular diagnostic companies and 2 clinical companies in the past, when I look at opportunities in companies where I'm going to spend my time, there are 4 elements that are important to me. First of all, I want what we do to have an impact. Well, in oncology, of course, that is very easy defendable. I like the approach to be different. One of the companies I worked for in 2000s, we used machine learning. Everybody is excited about AI, but we used machine learning to develop a gene expression-based algorithm to do breast cancer diagnostics. We took that company actually to the US. Dennis earlier talked about the US opportunity for healthcare, and I experienced that in Europe. It wasn't as good as it is here. So I took the company to the US, went to Irvine in 2008, and that is now a 250 people company with 80 million in revenue. So I look at opportunities that have an impact, and the biology needs to be different. I'd love to be in a clinical stage because I think clinical is the most interesting phase of the development, and that's personal. And I love some really solid biology behind it because, of course, nowadays it is all about the biology. So this opportunity with Lixte, I was on the board of Lixte since 22. And when the founder asked me to take over his role as CEO, I agreed because I think Lixte hits all those 4 elements of making an impact, being different, being in the clinical stage, and actually have a great, fascinating biology behind it. And so starting with the impact, we all know that in oncology, most cancer patients either get chemotherapy or immunotherapy. And, those drugs work relatively well, but earlier said already during the presentation, 10, 15, 20% response rates is probably optimistic, right? For immune, you've got a few exceptions, for example, melanoma and lung cancer, because those cancers have a lot of mutations. They're really different compared to healthy cells. And the problem with the immune system is that it's actually trained not to attack human cells, right? And a cancer cell for 99.9% is the same as a healthy cell. So the immune system really needs to be able to distinguish between the two, and it's not easy. And with lung cancer, particularly when caused by smoking, and melanoma caused by a lot of solar radiation, there's enough mistakes in the cell to actually for the immune system to recognize it. But all those other cancers, it doesn't work that well. So the immune system can't see the cancer well enough. And with chemotherapy, kind of the same. It is very toxic, so we can't dose as high as we want. So all patients today get either chemo or immune therapy, but we need to improve that. And so Lixte has a very interesting approach by developing a PP2A inhibitor. So this was initiated independent from Lixte. Lixte was founded in 2005, and they had a very different strategy. But my scientific friends at the Netherlands Cancer Institute, they were doing loads of screens in synthetic lethality, meaning if I add one drug to the other, when do I actually kill the cancer and it doesn't get up anymore? And they found that inhibiting the PP2A pathway was really interesting and lethal in certain combinations. So they said, let's look around the world if there's a pharma company already working on PP2A, because then we don't have to start from scratch. And they found Lixte. So they approached Lixte, and Lixte was very willing to collaborate. And we have a solid collaboration. Actually, we outsourced our R&D to the Netherlands Cancer Institute for that reason 2 years ago. And so why is this such an interesting and new approach? Because I said, besides the impact, the second element that I find is crucial is to be different. So if you inhibit PP2A, you actually activate cancer cells. Now that might sound counterintuitive initially, right? Because we always try to inhibit. If you look at all the pharma companies around the world, they are inhibiting cancer signaling, especially kinases, because a kinase is the on switch. So if I break or inhibit the on switch, then you don't have the signaling. And on paper, that's a great concept, and we've made great progress, and it does help. But very often the cancer still becomes resistant against those targeted therapies, right? And that's why, you know, we like immune therapy so much, but it doesn't always work. Those targeted therapies initially work, and then that cell becomes resistant. Now, we develop a phosphatase. The phosphatase is the off switch. So not only are we not breaking the on switch, we're actually taking away the off switch. So you get more signaling, which sounds completely counterintuitive. But in the preclinical models, it actually worked really well. And we really now understand why. We've done a lot of research. The company completely transitions from its original strategy into combination therapy for oncology for this reason, because we understand the biology. So what PP2A inhibition does is actually drive the cell into its division process. It starts to divide. And as we all know, we need dividing cells for chemotherapy to be effective. Chemotherapy is particularly effective in fast dividing cells, which we see when we look at, you know, losing your hair or your taste. Those are all fast dividing cells and a side effect of chemotherapy. So we drive cells in starting to divide. On the other end of the spectrum, we actually have an influence on the immune system. So into a little more detail later on the biology. So before I took over, the team did some great fundamental work. They done 2 phase 1 safety studies, because as I said, this approach is really different. And there was a big concern that it might be toxic on healthy cells, but it's not. It has very limited effect on healthy cells, proven by 2 completed safety studies for monotherapy. But I think most importantly, they did all the essential work to get a FDA IND approval to do clinical trials in the US. And they did all the work to get an IMPD approval to do clinical trials in Europe. We've outsourced our manufacturing. It is a relatively easy molecule to manufacture. And we've got sufficient GMP manufactured material to do a number of clinical trials. So the heavy lifting from that end has been done, and we're now entering the exciting phase of clinical trials and actually showing whether what we see preclinically translates to patients. So the mechanism, twofold. The mechanism of inhibiting PP2A on chemotherapy has 2 elements. One, it puts the cell into the dividing stage in cell cycle, as we call it, which makes it vulnerable for chemotherapy to actually make those effects that chemotherapy has on the dividing cell, disturbing the division and then driving the cell in apoptosis. And secondly, PP2A has an effect on DNA repair. So we break the DNA with the chemotherapy, and it's more sensitive for chemotherapy. But secondly, there is a mechanism to repair broken DNA, but that's also inhibited by the PP2A pathway, because this general phosphatase actually has a lot of targets in the cell. It gets even more exciting when we look at immunotherapy, because it has 3 effects in the case of immunotherapy, not only on the cancer cell, but also on the immune cells. So what we see is that inhibiting PP2A gets the T cell proliferation up. So you get more T cells, and that's what you want, because as you saw in the previous presentations, T cells are the ones that actually attack the cancer. But we also up the cytokine production. As I said, we take off the off switch, so there's more of everything. There's more cytokine, and that's the signal of the immune system to start doing something, right? And Thirdly, it has an effect on the cancer cell, which I will get to in more detail on the next slide, because we actually published that this week. This is still from our preprint in bioRxives, but the paper came out this week in EMBO, exactly showing how that mechanism works. So when you treat cells with PP2A, what happens is that when DNA is read and RNA is created, RNA needs to be spliced to be functional. And what PP2A does is actually disturb that process. And you get different parts of RNA that then translate into different proteins or different peptides. And those are we call neoantigens. These are pieces of proteins or proteins that the body doesn't recognize. And what we show in that article is that when that happens in the cell, they're actually also presented at the cell surface. And now the immune system has a bunch of red flags on the cell and makes it easier to recognize and attack. So inhibiting PP2A has an effect on both the immune cells with cytokine production and T cell proliferation, but also on the tumor cell by additional neoantigen creation, and therefore the immune cell can easily recognize it. Now, this is all phenomenal biology, but as I said, I'd like the clinical stage, right? So does this translate to patients? And that's the phase we're in with Lixte at the moment. So this is data without our molecule, but this is these are immune therapy trials. And what you see here, for those that are less familiar with Kaplan-Meier curves, you see that on day 1, 100% of the patients are alive. And the difference between the patients in the blue curve who do far worse than the patients in the red curve is that the patients with in the red curve have a so-called mutation in PP2A. So their PP2A gene is broken, and they respond much better to immune therapy than patients with a PP2A wild type. Unfortunately, the PP2A mutation is not that common in cancer, and therefore we take this approach of inhibiting PP2A with a drug, effectively creating more or less a PP2A mutant. And we don't see the same effect with other drugs in the other graph. So that brings us to 4 clinical trials currently, 1 planned and 3 are actually actively recruiting. 1 where we combine PP2A inhibition with our drug LB100 in soft tissue sarcoma. We do that with the Sarcoma Collaborative Trial Group in Madrid. We have a trial where we combine PP2A inhibition with LB100, our drug, with chemo and immune therapy, because that's the standard of care currently in small cell lung cancer with the City of Hope Hospital in Duarte. And we have a trial recently opened, and I'll go into a little more detail there, where we combine LB100 with immunotherapy in clear cell ovarian cancer. And that one just recently opened in collaboration with GlaxoSmithKline. And that was an interesting story. And then fourthly, that's planned where we are going to do a trial in metastatic colon cancer based on that neoantigen, because we know that immune therapy works really well in MSI high colon cancer. Those are highly mutagenic colon cancer tumors, but that's only 15%. The majority of colon cancer is MSI low. And what we think that we can do with LB100 is actually create a kind of an MSI high phenotype by treating with LB100. Going back to the ovarian cancer, the only one I will discuss in detail, but there's obviously much more information available on our website and in our filings, and feel free to reach out afterwards. In ovarian clear cell, this difference between having the PP2A mutation and being the wild type with just immunotherapy. So again, this is not without drug. This is a failed immunotherapy trial where after the fact, they looked at the DNA status of those patients and said, is there something in there that would still differentiate? And what you see is that all the responders, although only 7 out of the 21, but all the responders had that PP2A mutation. And so this is the rationale of using LB100 in clear cell ovarian cancer now in the wild type, because they don't have that benefit. And this was, again, very interesting. The way I got to this company through the Netherlands Cancer Institute, who cleared the biology and found out what fascinating biology there is behind Lixte. Here, MD Anderson actually came to us and to GlaxoSmithKline and said, we want to test this. And we were obviously open to do this. And Glaxo thought it was such a good idea that they actually picked up the bill to do this trial. So Glaxo pays for the trial and provides their PD-1 inhibitor, dostarlimab, and we provide the drug LB100. And so initially it's a phase 1b/2 trial, because as I said, LB100 is safe and an effective inhibitor of PP2A as monotherapy. But as soon as you start combining it with a new drug, you have to do an upfront toxicity evaluation. And so that's the 1b component of the trial, where we are upping the dose of both immune therapy and LB100 in combination. And then we'll continue running to see if we see an effect of the combination in this indication. And as I said, based on the genetics and the PP2A mutation, we think that this has a good chance to be successful. Coming to an end, concluding, as I said, I want to put my effort and energy into a company that makes a difference, that has an impact, that is clinical, that has strong biology. And so I think we are dealing here with a clinical stage pharmaceutical company and we're the only ones in the world that have a PP2A inhibitor in development. There is no other company on this pathway. It enhances the effect of both immunotherapy and chemotherapy. It's a safe and potent inhibitor of PP2A. And we currently have 3 trials open, combining PP2A with either chemotherapy or immunotherapy. And there's one more in the planning. And with that, I'd like to conclude and hope I entertained you for at least 15 minutes. There is another one, PP5, I think, that we have a suspicion that it will also inhibit. So it's pretty selective. I mean, we've done all the translational work to go there, but there is indeed, and that might be actually might be a benefit. But we go into great detail into our paper. So I'm more than happy to send you a copy of that.
There are tumor subtypes that have splicing deficiencies, that acquire splicing deficiencies that the drug may work better.
I haven't looked at it that way. So as the paper came out with the neoantigen, that was like for us the first time that we saw. So the interesting in the publication process, they really forced us to substantiate the claims to the point that we did phospholipidomics and really we had to put the evidence on the table that neoantigen is presented on the cell surface, right, on those MHAs. So we haven't looked in those areas specifically, but at least we know that hopefully it's a more generic way to do it, and it's not bound to limits, and therefore we picked MSI low colon cancer. Thank you very much.